guys. Today we are talking about connective tissue and blood, mainly connective tissue today. We have basic tissues in the body, which are four tissues: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. We will talk about connective tissue. The general character of connective tissue: it develops from the mesoderm, except that of head and neck from neural crest ectoderm. It means that all connective tissue in the body is developed from mesoderm except connective tissue presented in head and neck, which developed from neural crest ectoderm. Okay? Connective tissue presented under the epithelial tissue and separated from epithelial tissue and other tissues by basal lamina or basement membrane. Okay? Connective tissue is vascular while epithelial is avascular. Okay? Connective tissue vascular as it penetrated by blood vessels, lymphatics, and nerves. So, connective tissue mainly present in vascular tissues and responsible for nutrition of other tissues around it. Okay? What is the function of connective tissue? Connective tissue have many functions. Support, fat storage, medium for exchange between blood and tissues. As we said, it is responsible for nutrition of tissues around it. Okay? As or because it is a good medium for exchange between the blood and the tissues. It has a protective function as it contains a defense cell. Defense cells as macrophage, we will discuss it later. Repair the damaged tissue as fibrosis because it contains a cell called the fibroblast, we will talk about later also. So, function of connective tissue generally: support, fat storage. It's a good medium for exchange between blood and tissues, protection, and repair. Okay? Connective tissue have many types: connective tissue proper, embryonic connective tissue, specialized connective tissue. First, connective tissue proper. It's a type of connective tissue. Have many types: loose areolar connective tissue, dense collagenous connective tissue contain collagenous fiber, reticular connective tissue contain reticular fibers, adipose connective tissue contain fat cells, and elastic connective tissue contain mainly elastic fibers. The second type, embryonic connective tissue, have two types: mesenchymal and mucoid connective tissue. The last one is a specialized connective tissue, which is a special type of connective tissue and have a special character as blood, cartilage, and bone. Each of them contain its own special character. We will discuss each of these types separately. This is the mesenchyme, the origin of all connective tissue except head and the neck, as we said. This mesenchyme may be differentiated into fibroblast, which converted to fibrocyte. Fibrocyte forms connective tissue proper, which has many types, as we said. Mesenchyme can be differentiated to chondroblast, which can differentiate it to chondrocyte that forms the cartilage, and this type of the cartilage hyaline fibrocartilage elastic, and we will discuss it later. Mesenchyme may be differentiated into osteoblast. Osteoblast it will differentiate to osteocyte. Osteocyte which will form the bone. The bone contain compact and spongy bone, although we will discuss later. Mesenchyme also can be differentiated to hemopoietic stem cell that forms the blood cells, which will be RBCs, right, red blood cell, white blood cells, white BCs, and the platelet. We will discuss this in this module. What is the structure of connective tissue? Connective tissue structure, it's formed from three main parts: cells, extracellular matrix, and the fibers. Any connective tissue presented in the body is formed from these three parts: cells, matrix, and fibers. The cells, we will discuss it in this lecture. Okay, what about extracellular matrix? Extracellular matrix may be ground substance or tissue fluid. Tissue fluid is the fluid between the tissues. Okay, what is the ground substance in the connective tissue? Ground substance contains many substances, which may be GAGs, glycosaminoglycans, represented as GAGs, proteoglycans, which is protein and the glycoprotein and the glycogen, and the glycoproteins also. Okay. Fibers may be elastic, collagen, reticular fibers. This is the general structure of any connective tissue in the body. We will discuss today the cells. Cells of connective tissue may be branched, means it have a branches from the cell body, and unbranched, it contain no branches. Unbranched cell as unmesenchymal stem cell, undifferentiated mesenchymal stem cell, fibroblast, pericyte, macrophage, reticular cells, and the pigment cell. This is branched cells which contain branches. Okay, unbranched cell which contain no branches as mast cell, fat cell, plasma cell, extravasated leukocytes. We will discuss each of them now. Undifferentiated mesenchymal stem cell. It is called the mother cell of all cells of the body, as this cell will differentiate it to any cell of the body. So it's called the mother cell of the body. It may differentiate to endothelial cell, to smooth muscle cell, to osteoblast, which forms the bone, to chondrocyte or chondroblast, which convert to chondrocyte that forms the cartilage, and the adipocyte that forms the fats. Okay, so this is called mother cell of the body, which is the unmesenchymal stem cell. This is the cell body, and from which the extended process or branches, so it is one of the branched cells. Other cell is called the pericyte. Pericyte, the cell, it's a cell presented in the wall of the vessels mainly, as the basement membrane of the blood vessel splits to enclose pericyte like that. This is the basement membrane. We said that basement membrane from here it's split to be two membranes. In between the two parts of the basement membrane there is a cell. This is called the pericyte. So pericyte is between the two splitted parts of the basement membrane. What is the function of this pericyte? Pericyte has two functions. It's a contractile cell that regulates the flow in the capillary. Why? The capillary, it is the site between artery and vein. Artery and vein contain different blood pressures. When the blood passes from one of them to the another, it passes to a different pressure. So it needs a push to be passed through the another blood. This push, due to contraction of the pericyte cell in between the two splitted parts of the basement membrane, so it regulates the flow of the blood in the capillary. The another function, it acts like a stem cell. It acts as a stem cell that regenerates the endothelial cell. We know that stem cells regenerate any cells inside this organ. So pericyte act as a stem cell that regenerates the endothelial cell. What is the endothelial cell? Endothelial cell, it is a single layer of flat cell that lines the lumen of the blood vessel. This is called endothelial cell. This pericyte is responsible for regeneration of endothelial cell if it has any injury. This is the pericyte cell. Another cell, this is a fibroblast. Fibroblast, it is the most common cell of connective tissue. Fibroblast, again, it is the most common cell of the connective tissue. Fibroblast contain branches as it is a one of the branched cell. Okay, fibroblast presented as two types. It may be presented as fibroblast or fibrocyte. What is the difference? Fibroblast is young and active. Fibrocyte is old and inactive. So if fibroblast became old and inactive, it transferred to it became fibrocyte, which is old and inactive. The character of fibroblast, it is responsible for synthesis of connective tissue fibers and the matrix. As we said, connective tissue, any connective tissue mainly contain cells, fibers, and the matrix. The cell which is responsible for synthesis of fibers and the matrix is the fibroblast, as it is the most common cell of connective tissue. Although it is responsible for connective tissue growth and repair. So, fibroblast responsible for connective tissue, synthesis of connective tissue fibers and the matrix, and growth and repair of connective tissue. This process of repair is called the fibrosis from the general function. Fibrosis, it's a type of repair occurred by fibroblast. Okay, fibrocyte. Fibrocyte, it reverted to active fibroblast in wound healing. As we said, that fibrocyte as a fibrocyte is inactive. So, what is their function? Their function as a fibrocyte, it has no function because it is an inactive. So, if there is a wound healing and I need a fast process of or fast fibroblast to repair my injury, fibrocyte will revert to the active fibroblast in wound healing to facilitate the healing of the wound. Okay, this is the fibroblast and the fibrocyte. This is active one and this is inactive one, young one and old one. This cell revert to fibroblast in case of wound healing to facilitate this process. Okay. What is the myofibroblast? Myo from muscle. It is a cell with both features of fibroblast and the muscle cell. So it is called myofibroblast. It has the characters of muscle and the fibroblast. It is responsible for wound healing and tooth eruptions in babies. Okay, so myofibroblast have the characters of smooth muscles and the fibroblast and responsible for wound healing also and tooth eruption in infants. Clinical application for this cell or fibroblast cell or specifically in fibrosis or repair. Hypertrophic scar or palmar fibromatosis. It occurs by second week of wound healing. The most of the myofibroblast undergoes apoptosis and disappear. My wound is healed by the second week. So myofibroblast, I don't need it anymore. So it will undergo apoptosis and disappear. But what happened if the myofibroblast persists? It causes a formation of more fibers. So it causes hypertrophic scar like that. Hypertrophic scar formation resulting in excessive connective tissue contraction and leads to deformity. Okay? Another cell, macrophage. Macrophage is the defense cell of the connective tissue. It may be fixed cell or free cell. Fixed presented in connective tissue only, so it is called the fixed. But free one it migrates to another tissue. So if the macrophage presented only in connective tissue and doesn't migrate or move to another tissue is called the fixed cell. But if it is migrate to another tissue, this is called the free macrophage. Okay, macrophage cell have many names. Name of macrophage differs according to the site of macrophage. Macrophage of the tissue, of any tissue, connective tissue, epithelial tissue, is called the histiocyte, as histo means the tissue. But connective tissue of the liver called von Kupffer cell. Connective tissue of the lung called dust cell, as it is macrophagocytose the dust from the air. Macrophage of the skin is called the Langerhans cell. Macrophage is the brain is called the microglia. So all of them is a macrophage, but macrophage have all of their different names according to the site. What is the function of macrophage? Macrophage is responsible for phagocytosis of any foreign body in all of the organs. So it is the defense cell or defense function of connective tissue. Another function, immunity. Why immunity? Because it is act as antigen presenting cell. It is present the antigen to the immune cells. So it has immunity function. Last one, it has a secretory function. Secretory function as it secrete what? Secrete cytokines, chemotactic factors, and collagenase enzyme, which also have a defense mechanism. So the main function in all of this is defense. Okay, this is electron microscope of macrophage. Contain many lysosomes, as the lysosome is the organelle in the cell presented in phagocytic cells. and the kidney, and the nucleus, which is kidney-shaped nucleus, okay, and pseudopodia, which is a layer fruits called pseudopodia. This is electron microscope, and this is light microscope stained by vital stains as trypan blue. Another cell is a pigment cell. Pigment cell presented in pigmented areas. Pigmented area. What is our pigment? Our pigment is called the melanin. So, pigment cell presented in colored part. What is the colored part in our body? Skin, hair, and eyes. These three parts is a colored parts and contain melanin pigment. Melanin pigment is secreted from a pigment cell. This pigment cell is called the melanophores in animals and the melanocyte in mammals. Mammals like humans. So melanophores in animals and the melanocyte in mammals. This is a two names of the same cell that is which is a pigment cell that secrete melanin pigment in our skin, eyes, and hair. Another cell is the mast cell. Mast cell, it is the allergy cell, which is responsible for allergic reaction. Mast cell have a role in allergy by synthesis, storage, and release of mediators of allergy. So mast cell, it is the allergy cell as it synthesize, store, and release of these mediators of allergy. What is the mediators of allergy? We have heparin, sulfated, which contains sulfated gags, histamine, ECF stands for eosinophil chemotactic factor, NCF neutrophil chemotactic factor, SRSA slow reacting substance of anaphylaxis. All of them is the mediators of allergy that stored in the synthesized and stored in mast cell and released in case of allergy, sorry. So what happened if we exposed to allergy or allergic antigen? On first exposure to the antigen stimulate beta lymphocyte or P lymphocyte to be activated B cell that forms the plasma cell. Plasma cell forms the antibodies, mainly IgE. It is the antibody responsible for allergy that bind to its specific receptor in mast cell. So this antibody binds to mast cell on its specific receptor, which leads to formation of sensitized mast cell. Sensitized mast cell is the mast cell that's sensitive to this antigen. But what happened if we exposed again to the same antigen? The antigen will bind to the IgE on this mast cell. Actually, on this sensitized mast cell. Sensitized mast cell is a mast cell contain IgE. The antigen will bind to this cell, leads to degranulation of its granules. Its granules contain these mediators, which leads to release of the mediator and present as a manifestation of allergy. So allergic reaction doesn't happen from the first exposure, it happened from the second one. First exposure leads to only a sensitized mast cell, which is a mast cell contain IgE antibody. If we expose it to the same antigen will attach it to this sensitized mast cell leads to its degranulation and release of its granules containing mediators. These mediators will appear as a manifestation of allergy. Like that, this is the mast cell. This is the sensitized mast cell, which means the mast cell contain IgE antibody on its surface. The blue one is the receptor and the red one is the antibody. So antibody attached to the receptor on mast cell, this cell is called sensitized mast cell. So the first exposure is done. If the antigen attached to receptor, there is degranulation of the mast cell and release of the mediators of allergy, and the manifestation of allergy will appear. The light microscope of the mast cell doesn't stand by H and E, but it will stand metachromatically by toluidine blue. Metachromatically means the change in color. So mast cell stand metachromatically by toluidine blue stain. This is the mast cell. Another picture, mast cell stained metachromatically by toluidine blue. This is the electron microscope of mast cell, which contain many granules contain allergy mediators. This is all of these vesicles around the cell is granules. These granules contain allergic mediators. Okay, what is this on the surface? This is the receptor for IgE. So mast cell contain mainly inside the cell many or numerous granules which contain allergy mediators, and on its surface it contains a receptor for the IgE antibody. This is the manifestation of allergy, which is called hypersensitivity reaction type 1. It's the type of the allergy, as allergy contain many types of hypersensitivity, actually 4 types. This is a hypersensitivity reaction type 1, appears as asthma in the lung, bronchial asthma in the lung, or hives in the skin, or food allergy, which appears as enlargement in the lips, or eczema like that in the skin. This is the manifestation of the hypersensitivity reaction type 1 mediated by mast cell and the allergic mediators. Another cell is plasma cell. Plasma cell formed in the pathway in the formation of sensitized P and sensitized mast cell, as plasma cell formed from P cell and give antibody. Plasma cell formed from P lymphocyte, so P lymphocyte forms plasma cell. Plasma cell forms antibody, so it forms from B cell and gives antibody. By light microscope, plasma cell appears with the most special character, which is negative Golgi image. Negative Golgi image, it is a pale area around the nucleus. Supranuclear area, pale area. This is called negative Golgi image. It's the most characteristic feature for light microscope of plasma cell. طيب. What about electron microscope? Electron microscope, it contains peripheral chromatin. This peripheral chromatin arranged inside the nucleus in a clock face manner or cartwheel appearance. So electron microscope of plasma cell have the most characteristic features, which is the arrangement of peripheral chromatin inside the nucleus, which is clock face or cartwheel appearance. Another cell which is flat cell. Flat cell, fat cell, sorry. Fat cell may be unilocular adipocyte or multilocular adipocyte. What does that mean? Unilocular adipocyte or white fat cell means this one large granule of fat. This type of cells presented under the skin around internal organs like kidney and behind the eyeball. What is the function? The function of white fat cell is synthesis and storage of fat, mainly triglyceride, heat insulation in cold weather, supporting function as fat around the kidney. As we said, it presented around the internal organs, so it is responsible for support for the organs, mainly kidney. Okay, this is the white fat cell or unilocular fat cell, one granule of fat. Okay, what about multilocular? Multilocular or brown fat presented as many granules of fat. So there one granule and there many granules, so it's called the multilocular adipocyte. It's presented mainly in the newborn, at neck, axilla, and the interscapular region. The main function is heat generation to produce heat in the babies. This is white fat cell, which is unilocular. It contains one large granule of fat that store energy. And this is brown fat or multilocular fat, which contains many small granules of fat which burn energy. And this is the nucleus, and this is the mitochondria that generate fat, and this is fat droplets. One granule, unilocular, multi-granules, multilocular, white fat, brown fat. So why brown fat is stained brown, or it's why is the brown color of the, what's the cause of brown color of brown fat? Because brown fat is high vascular, contain high vascular supply and high number of mitochondria rich in cytochrome enzyme. So brown fat is brown in color due to high vascular supply and high number of mitochondria rich in cytochrome enzymes. This is responsible for the brown color of the multilocular fat. H&E staining, this is fat droplets, appears as empty granules. These empty granules makes the side ring appearance. Why? Because fat is dissolved during the preparation of the slide. So appears like that is fat cells. This is one large fat droplet. So this is the unilocular fat or white fat. This is multilocular fat droplets. So this is the brown fat or multilocular fat or multilocular adipocyte. Special stain of fat may be Sudan 3, which appears orange in color, or Sudan black, which appears black in color. The electron microscope of multilocular fat appears like that. There is many small granules of fat droplets presented inside the cell, white in color. So this is the multilocular adipocyte or brown fat, which appears as multiple large or multiple small granules of fat presented inside the cell. And that's it for today, and have a nice day, guys. Thank you.